Myosin II ATPase is a hydrolase enzyme that takes part in muscle contraction. We will be presenting an article that is studying the mechanism by which this occurs and presents a novel hypothesis on the mechanism. Myosin II ATPase is a kinase protein that belongs to the superfamily myosin. This protein binds actin filaments and pushes it, or rows it. This movement of the actin filaments causes muscle contractions that are used for movement. When myosin binds to the actin, there is a conformational change of the myosin head, causing it to alternate between a 90-degree relative to the neck and a 45-degree orientation. In other words, the head binds the actin filament while at a 90-degree orientation, and the ATP binds the myosin head, is hydrolyzed to ADP, and this causes the head bound to the actin to switch to a 45-degree orientation. This change pushes the filament before the head quickly unbinds, and a new ATP molecule binds to the myosin head, starting the cycle again.
Myosin II ATPase is a hydrolase, part of the myosin-kinsin superfamily of proteins and of the family myosin. Its function is to hydrolyze ATP, bind to actin, and transduce mechanical energy. This protein is composed of 2 heavy chains and multiple light chains, organized into 3 structurally and functionally different domains: the head, neck, and tail. The head is composed of heavy chain. The head functions as the location of actin and ATP binding sites and generates the force required for the conformational change that propels the myosin along an actin filament and thereby acting as the ATPase. This domain corresponds to the end terminus of a heavy chain.
The neck is composed of light chains. Both regulatory and essential domains regulate the activity of the head domain. The third domain, the tail, determines the role of each protein in the myosin family. In myosin II, the tail is composed of heavy chains that form a coiled coil structure motif, resulting in relatively long, rod-like structure. Multiple molecules of myosin II will pack together by means of their tail domains to form a thick filament. Here, the tail domains form one large rod with multiple head domains protruding, able to react with the actin filament. This domain corresponds to the C-terminus of the heavy chain.
Protease cleavage has been used to determine the full domain structure of myosin. Typically, proteases cleave myosin II at the base of the neck domain, producing a rod-like tail fragment called the light meromyosin, and a head and neck chain called the heavy meromyosin. Further digestion is required to separate the head and neck domains. Reacting the heavy meromyosin with a papain separates the neck domain from the head domain and isolates the 2 heads of the molecule.
Myosin II is a hexameric protein. A simple description of the structure of this protein has the head domain at one end, with the N-terminus ends of the heavy chain intertwined, with the 4 light chains that lead into the neck of the protein. Extending from this bundle of chains is the coiled coil structure of the 2 heavy chains forming the tail domain. The end of this tail has a shorter end at a different angle. This is part of the tail that binds to the actin and will transition between 90 and 45-degree angle. Myosin II has 3 major parts: the S1 or head domain, which is the binding site for the actin filaments and the ATP, made of heavy chains. It has the neck, made of light chains, and the tail composed of heavy chain. There are 2 heavy chains in each myosin II molecule that each comprise a head connected to 1 coil in the tail.
There are 2 kinds of light chains in myosin II, and 2 of each are located at the base of the myosin head: regulatory and essential. Regulatory chains function in calcium 2+ binding, stiffening the neck and preventing a conformational change of the myosin head. However, the exact method of this function is unknown. It has been determined that serine 19 on the light chain is phosphorylated, causing the head to contract and the neck to stiffen, preventing binding. Phosphate kinase regulates this process by dephosphorylating the regulatory subunit. As such, essential chains are less well understood. They participate in calcium 2+ binding as well, and they are suspected to stabilize the myosin head somehow. It is suspected that myosin bind cooperatively, meaning that bound myosin heads heighten the affinity of nearby heads for the actin filament. In low ATP environments, this tendency causes the heads to bind in clumps on the actin, leaving other parts bare. This reflects the role of favorable conformation of actin filaments on the relative affinity of binding myosin heads. However, this variability is difficult to detect in vivo due to low affinity and the presence of ATP.
The Limb-Taylor cycle, pictured above, is the mechanism describing the function of myosin. Actin acts as a stimulant and a nucleotide exchange factor for myosin II, stimulating the hydrolyzation of ATP to form ADP, providing energy for the conformational change of the molecule. Myosin forms a cross bridge that is bound to the actin in the rigor or 45-degree down position. At this stage, the active site is considered open. ATP binds to the ATPase site on myosin, quickly dissociating myosin from the actin and changing from the open to closed position. Hydrolysis of the ATP to produce ADP, an inorganic phosphate, leads to the return of the myosin crossbridge to the 90-degree up position, where it rebinds the actin at a new location on the filament. This causes the release of the ADP and returns the myosin head to the 45-degree state, rowing the actin, which allows the myosin to again bind a new site on the filament. Myosin II ATPase, expressed by the gene commonly denoted as MYH2, contains 1,942 amino acid base pairs that are localized on chromosome 17 in humans. These amino acids make up 6 peptide chains that join to form the protein. Myosin II has a similar amino acid sequence to multiple proteins in the myosin family. It is very closely related to myosin I in the number and species of amino acids in its peptide chain. Myosin II contains 1,941 amino acids, and myosin I contains 1,939. Between the two, 95% of the identities, meaning the location and the type of amino acid, are identical. Myosin V, on the other hand, contains only 1,219 amino acids, and only identifies with 35% of the amino acid residues in myosin II.
There are many known splice variants of our enzyme, including but not limited to non-muscle myosin II A, B, and C. The major difference between these splice variants are the location in which they are expressed. No tissue or cell type has been shown to express all 3 of these types. Structurally related proteins, such as those others in the myosin family, perform a similar function to myosin II. Myosins interact with actin filaments as motor proteins. Muscle myosins contract muscles through this interaction, binding to the actin through their head domain. Different myosin genes are composed of differing amounts of heavy and light chains, but form the same basic domains of head, neck, and tail. For example, myosin I has very similar mRNA structure to myosin II, and has a head and neck domain, but it is lacking a tail domain. This protein also participates in muscle contraction, actin filament binding, and is therefore considered a motor protein.
Fully understanding the mechanism of myosin II ATPase and its binding and unbinding to actin will give great insight to some commonalities within the superfamily. The different types of myosin share many structural analogs and is therefore assumed to share several mechanistic steps. Actin filaments are linear polymers of globular actin, found as microfilaments in the cytoskeleton and thin filaments in muscle and non-muscle cells. Within muscle cells, they are aligned for the myosin proteins to push the filaments, known as thin filaments, along, causing muscle contraction.
The study we are presenting utilized GFP, a green fluorescent protein in jellyfish, as a stain to visualize infused myosin heads with amino acid substitutions that would heighten the affinity for actin filament binding in the presence of ATP. The goal being to identify subsets of actin that myosin II would preferentially bind to. They were successful in discovering myosin heads preferred mechanically stretched sections of the actin filament, while in vivo conditions existed.
The soil-dwelling amoeba Dictyostelium discoideum was used for this study. The cell was shocked, opening up pores in the cell membrane and allowing the stains to infiltrate the membrane and express slightly different versions of the myosin heavy chains. The medium used to house the cell culture was HL5. Utilizing confocal fluorescent microscopy, My cell observations were made of the GFP mutant head and the GFP mutant myosin II heavy chain. Cells were allowed to settle to the bottom of the Petri dish and scanned with a confocal laser scanning unit. This method of microscopy scans point by point, allowing multiple depth viewing and thereby a three-dimensional rendering of the sample. Cells were starved for 8 to 10 hours to induce chemotaxis. This is important for stimulation of the amoeba to move towards something, i.e. food, allowing the viewing of the actin stretching.
This same method was used to observe fixed cells on glass bottom dishes, made permeable and fixed by replacing potassium sodium phosphate buffer with a solution of pipes, magnesium chloride, EGTA, DTT, Triton X100, and glutaraldehyde. After fixing cells for 10 minutes, they were stained for 1 hour in PBS with RH-pH stain, rinsed in PBS containing DTT, and observed through confocal laser scanning microscopy. 2 types of stains, or probes, were needed to detect actin filaments with higher affinity for myosin II motor domain. One that would accurately report local concentration of actin filaments, and one that would preferentially bind to subsets of actin filaments having higher affinity for myosin II motor domain.
In the report of local concentration of actin filaments, the stains compared were GFP actin binding domain of actin binding proteins, and RH-pH. GFP actin and RH-pH were found to be very similar, while GFP-ABD and RH-pH were very dissimilar. RH-pH was then chosen as the actin stain for the study, as it allowed for total actin filament concentration to be semi-quantified. As for the preferential binding of subsets of actin filaments with a higher affinity for the myosin II motor domain, the use of fluorescently labeled motor domains lacking a tail domain was required. This was done because filament formation involving the tail domain may localize myosin II filaments in vivo. At physiological levels of ATP, the head only weakly associates with the actin filaments.
This mechanism is demonstrated by the limb-Taylor cycle, a slow actin-stimulated release of ADP, an inorganic phosphate from the head, establishes a strong binding with the actin filament, followed by a rapid power stroke, the rowing action, that is almost immediately followed by the dissociation of the actin filament from the head. The amount of time that the myosin head is actually bound to the actin filament is very short, and for this reason, a mutant Myosin II ATPase, designated G680A, was incorporated into the head of the proteins fused N-terminally to GFP. This mutant is one of many that is made with an extended strongly bound state in the presence of ATP. These trials found that both myosin head mutants showed unique localizations along the actomyosin cortex, particularly around the front and rear of the cortex. From this, it was inferred that the GFP S1 mutant stained myosin II head could provide consistent results and was therefore chosen for the probe required to show affinity higher than naturally occurring myosin II head.
Mutant cells were permeabilized and fixed in the presence of Triton X100 and glutaraldehyde, then stained with RH-pH and GFP, and the resulting fluorescence appeared similar in all cells examined. A comparison of patterns between the two stains showed reproducible differences in the distribution of bound S1. Figure 4 here displays the differently stained samples and their signal densities. A shows a multinucleated P10 phosphate-intensin homolog undergoing cytokinesis. The far left image is a GFP stained fluorescence image, the middle an RH-pH stained fluorescence image, and the right image is a superimposition of the two. B and C display mutated cells undergoing cytokinesis. Preferential binding of certain regions of the actin filament to the myosin II is visible in the section when compared to A, instead of the evenly dispersed coloration seen in A. These cells show spots of coloration, with regions along the actin filament left gray and uncolored, showing regions without any myosin head bound.
This paper introduces the proposed mechanisms for the preferential binding of myosin II to actin filament. Myosin transiently forms bipolar filaments and associates with specific subsets of actin filaments to drive local contraction of the cortex. Myosin II filaments must selectively bind these subunits of actin filaments. The first proposed mechanism begins with a mechanosensor triggering a biochemical pathway, leading to enhanced affinity of actin filaments for myosin II. Isoforms of trypomyosin have been shown to differently regulate affinities of actin filaments for different classes of myosin heads. However, the genes for this protein do not exist in the genome for the organism used in this study, making this speculation irrelevant for findings in this experiment.
The second proposed mechanism assumes that during cytokinesis, a stretch is induced upon the actin cytoskeleton, aligning the filament for binding with myosin II. In the absence of ATP, myosin tends to form bundles of actin filaments. Therefore, it is possible that GFPS1, carrying a mutation heightening its affinity for the actin in the presence of ATP, prefers binding to the aligned or stretched areas of the actin filaments. Upon the cell receiving a mechanical stimulus and beginning to move, the filaments are stretched, causing a conformational change in some of the actin subunits.